All right, today we're tackling a really fascinating genetic puzzle drawing from the book The Origins of Schizophrenia. We are going to explore one of the most complex and, honestly, one of the most interesting questions in modern medicine.
And here's the question at the heart of it all. If we know schizophrenia has a strong genetic component, and it can sometimes make it less likely for a person to have kids, then why is it still around? From a strictly evolutionary point of view, shouldn't natural selection have weeded it out by now?
Well, this exact puzzle actually has a name. It's called the Darwinian paradox. And it's the perfect way for us to jump into the modern genetics of schizophrenia, because the answer, as you'll see, is anything but simple, but it's incredibly revealing.
Okay, so here's how we're going to break this down. First, we'll look at the puzzle itself. Then we'll go hunting for common genes. After that, we'll explore what happens when DNA gets copied or deleted. Then we'll get into something called the gene switch. And finally, we'll pull it all together. Okay, first up, we're going on a hunt for what are called common genes. This is really the first major piece of the puzzle, where scientists launched this massive, data-heavy search across the entire human genome looking for tiny, subtle differences that might add up.
So to really get this, think of it like this. Imagine the human genome is a gigantic library with billions of letters in its books. For this first step, researchers aren't looking for a missing chapter or anything that dramatic. Nope, they're looking for incredibly common, single-letter spelling errors, little typos that just happen to show up a bit more often in people with schizophrenia.
And the tool scientists use for this massive hunt? It's pretty revolutionary. It's called a genome-wide association study, or GWAS for short. It's basically this powerful super scanner that lets them compare the genomes of thousands upon thousands of people all at once to pinpoint these tiny genetic variations.
And this is what the results look like. It's called a Manhattan plot, and you can totally see why, right? It looks like a city skyline. Every single dot you see is one of those genetic typos. The really high skyscrapers are the ones that are statistically significant. They're like giant flashing signs telling scientists, hey, look over here. This part of the genome is important.
So what's one of the biggest skyscrapers they found? Well, it points to a spot on chromosome 6 called the major histocompatibility complex, or MHC. And this is where it gets really interesting, because the MHC is a huge player in our immune system. This was a massive clue, suggesting a direct link between our genes and our environment. Like maybe an infection during pregnancy could interact with a person's genetic risk. It's a key piece in solving that Darwinian paradox.
But the story doesn't stop there. Not even close. The genetic story gets way, way bigger than just tiny common typos.
Okay, back to our library analogy for a second. We've been looking for tiny spelling errors, right? Things that are pretty subtle.
Well, now we're talking about something else entirely, like whole paragraphs or even entire pages of the genetic code just being ripped out or accidentally copied and pasted twice.
So, in the world of genetics, these are called copy number variations, or CNVs. And you've got to understand, these are not small changes. A CNV can be a huge chunk of DNA, sometimes covering dozens of genes. It's a major, major structural change.
This is a great way to see the difference. On the left, everything's normal, but on the right, that word quick got duplicated. That's a CNV. It's a much bigger disruption to the sentence than a single typo would be, and as you can imagine, it can have a much bigger effect.
Now, here's the really important part. These big structural changes, they're rare, way rarer than the common typos. But when they do happen, their impact on an individual's risk can be massive. And because they're often brand new mutations, they can just pop up, totally bypassing that slow and steady process of natural selection.
All right, now for our third and final layer. And honestly, this one is a total game changer. Because it turns out, it's not just about what genes you have, it's about which ones are actually turned on or off. Let's stick with that analogy of a script, okay? If our DNA is the script of a play, with all the lines written out, what tells the actors which lines to deliver, which ones to say loudly, and which ones to skip entirely? Well, that would be the director's notes.
That's where epigenetics comes in. It literally means on top of the genome. It's like a whole other layer of information sitting on our DNA. These little chemical tags don't change the script itself, but they control how it gets read. You can think of it like the software that tells the hardware of our genes what to do.
So how does it do this? Well, the director has a couple of main tools. One is called DNA methylation. This usually adds a chemical tag that acts like a silencer, just turning a gene off. The other is histone modification,
which controls how tightly the DNA is wound. If it's loose, the gene is on. If it's wound up tight, it's off.
And here it is. This is the part that connects everything. This epigenetic layer isn't set in stone. It's dynamic. It can be changed by our life experiences. Things like stress, nutrition, or infections can all leave their mark. This is how our environment can literally talk to our genes, and it's a huge piece of that Darwinian puzzle. A person could carry genetic risk their whole life, but if it's never switched on by the environment, it has no effect and just gets passed on silently.
Okay, so we've looked at these 3 totally different layers of the genetic story. Let's pull it all together now and see how this complex mosaic finally gives us an answer.
It really is like a mosaic, and it gives us a pretty powerful answer to that Darwinian paradox we started with. First, you've got the common spelling errors. Since each one adds just a teeny tiny bit of risk, they can easily fly under evolution's radar. Second, the rare deleted paragraphs, the CNVs. These can just appear as new mutations, so they aren't always inherited. And third, you have the director's notes, the epigenetics, which means someone can carry risk genes that are never switched on, allowing them to pass them down.
And this brings us right back to a key point from the authors of our source material, Alan Brown and Paul Patterson. They say it perfectly. The origins almost certainly involve both genetic and environmental factors. It's not nature versus nurture. It's nature working hand in hand with nurture.
So if there's one thing you take away from all this, let it be this. There is no single gene for schizophrenia. That idea is just outdated. The genetic risk isn't one broken part. It's a really complex system of vulnerabilities spread out across our entire genome, constantly interacting with the world around us.
And that really is where the research is headed now. It leaves us with a huge, forward-looking question. Now that we understand how incredibly complex this genetic picture is, how do we take that knowledge, that amazing map of risk, and use it to develop real, effective treatments? And maybe, one day, even a cure.